carbohydrate, the rest of the part of previous lecture, in lecture 9. Okay, so what are polysaccharides? Polysaccharides are polymer of monosaccharide. Let me tell it glucose or yes, basically glucose. Okay, so there are two types of polysaccharide. One is homopolysaccharide, another is heteropolysaccharide. So homopolysaccharide contain only a single type of monomeric unit, that means glucose, 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 glucose. But in heteropolysaccharide, this contains two or more different kind of monomeric unit. So here is the structure of homopolysaccharide, and here is the structure of heteropolysaccharide. So heteropolysaccharide might composed of one, two, or several different monosaccharide units in a straight or branch chain. Okay, so homopolysaccharide can also be unbranched or branched like heteropolysaccharide. So what are the examples? So homopolysaccharide mean the example of homopolysaccharide includes glycogen in animals, starch and cellulose in plants, and heteropolysaccharide, the peptidoglycans of bacterial cell wall. Okay, so here you can see the structure of starch, glycogen and cellulose. So starch are moderately branched, and glycogen is highly branched, and cellulose is unbranched. So starch are two types. One is amylose and amylopectin. Amylose contain no branch, amylopectin having branched, glycogen containing severe branch than amylopectin, and cellulose doesn't contain any branch. And here are the types of bonds found in each types of macromolecules. Let's say in case of amylose, the monosaccharide units are glucose, only alpha glucose. They make alpha 1 full linkage. This is alpha 1 full linkage, alpha 1 full linkage, alpha 1 full linkage. And starch, the another part of starch is amylopectin, that is composed of glucose, that is alpha glucose, and contains both alpha 1,4 and alpha 6 linkage. This is alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4, but this is alpha 1,6 linkage. Okay, and in glycogen, starch, in glycogen, this is also composed of alpha glucose, contains both alpha 1,4 and alpha 1,6 linkage. And cellulose, they are polymer of beta glucose, so they can contain beta 1,4 linkage. Okay, so most of the animal cannot use cellulose as a fuel source because they lack an enzyme to hydrolyze beta 1,4 linkage. So the only vertebrates that are able to use cellulose as food are cattle and other ruminants like sheep, goats, camel, giraffes. The extra stomach compartment called rumen of the ruminant teems with bacteria and protists that secrete cellulase. This is an enzyme that can digest cellulose. So this is a homopolysaccharide chitin. Chitin is a linear homopolysaccharide composed of N-acetylglucosamine. So this is the structure of N-acetylglucosamine. In class, I have already shown how to draw the structure of N-acetylglucosamine. To summarize again, so firstly, we can say N-acetylglucosamine. So firstly, this is a glucose and amine, derivatives of glucose. So firstly, write down the glucose. And this is obviously beta D glucose. So the hydroxyl group at the anomeric carbon is upper side, so this is a beta glucose. So write down the glucose, beta glucose structure, and in number position 2 it will be changed. So glucose amine, that means number 2 position hydroxyl group will be replaced by amine, then it is called glucosamine. And when the amine group will be changed to acetyl group, then it will be called N-acetylglucosamine. And this is the monomeric form or monomeric unit of chitin. So the only chemical difference from cellulose is the replacement of hydroxyl group at C2 position with the acetylated amino group, and chitin form extended fibers similar to those cellulose, and like cellulose cannot be digested by vertebrates. Chitin is the principal component of hard exoskeletons of nearly a million of species of arthropods like insect, lobsters, crabs, for example, and is probably the second most abundant polysaccharide next to the cellulose in nature. So this is a beta-1,4 linkage. So monomeric unit, N-acetylglucosamine, 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 they are combined together by beta-1,4 linkage. And bacterial and algal cell will contain structural heteropolysaccharide, and peptidoglycan is a polymer consisting of a sugar and amino acid that form a mesh-like layer outside the plasma membrane of most bacteria, forming the cell wall. The sugar component consists of alternating units of beta-1,4 linked N-acetylglucosamine and N-acetylmuraminic. So this is the structure of N-acetylglucosamine, and this is the structure of N-acetylmuraminic acid. So after writing N-acetylglucosamine, the number 3 position, the hydroxyl group, will be replaced by acetylated muramic group. So these are two different monosaccharide units combined to produce heteropolysaccharide. So here are the look at here, this is the N-acetylglucosamine, this is the N-acetylmuraminic acid, and these are pentaglycine cross-link. That means these are peptide structure. Okay, so what are the glycoconjugates? Glycoconjugates is the general classification of carbohydrate covalently linked with other chemical species like proteins, peptides, and lipids. And there are 3 types of glycoconjugates. One is proteoglycan, second is glycoprotein, third is glycolipid. So proteoglycan, these are macromolecules of cell surface or extracellular matrix in which one or more glycosaminoglycan chain are joined covalently to membrane protein or a secreted protein. They are the major components of connective tissue such as cartilage. Aggregans is the most abundant proteoglycan in cartilage. Glycoproteins, they have one or several oligosaccharides of varying complexity joined covalently to a protein. For example, mucin, that is secreted in the mucus of respiratory tract and digestive tract. Another example is collagen. And glycolipids are lipids with a carbohydrate attached by a glycosidic bond, and their role is to maintain stability of the cell membrane and facilitate cellular recognition. So glycoprotein means protein attached with oligosaccharide. Glycolipids means lipid attached with carbohydrate, and proteoglycans mean protein attached with glycans. So oligosaccharide means short-term sugar chain, and glycans mean long chain length, sugar chain. Okay, so you can see here the proteoglycan structure. So this is protein and proteoglycan sugar chain. Okay, so that's all from this lecture.